Okay, there it goes. So if you could please just type in your name, title, tribe, and email in the chat, that would be great. So we can get any information out to you as it comes along and get everybody's in our contact list through our listserv. So not Mr. Dr. Miller, if you would like, that would be great. All right, Colton, can you share your screen? Is everything working for you?
Yeah, I'm here.
Okay, got it. I can see it.
So welcome back, everyone. I'm going to post the link to the Google Drive in the chat, and I can post it again after, as it will probably get filtered through everyone's introductions here. But just as a reminder, I've updated some of the materials on the Google Drive, mainly to add an additional deck of slides, an exercise, and the schedule for today. I've tried to cut things down a little bit, although you'll see when we go through the graphing stuff, there's essentially there's a ton of graphing resources and approaches to different ways to graph things in R. So hopefully it's not too overwhelming. Trying to strike a balance between providing enough complexity in examples of different styles of graph and ways to manipulate the elements and display of those graphs and kind of not overwhelm. But this way, not only can we walk through and kind of see and make ourselves aware of the functionality and like potential of R for doing this work, but also have this exercise as a resource to refer back to when, you know, later on you get to working with your own data and you can recall seeing or doing some of this, but not exactly what the codes or commands were. And you can look at the exercise and see, you know, line by line exactly what we did and refresh yourself that way. So bear with us as some of it might get somewhat tedious as we're going through it, but hopefully, given that these are, you know, real world data sets that I, gathered online, we can, you know, see some insights about them and keep engaged in that way. This is the tentative schedule for today. I thought that we could just start by kind of finishing the exercise that we were working on last time, as you know, as quickly as we can. I don't think it will take too much time to work back through that bit. And essentially, if you were, you know, doing that alongside, you could just follow along at this point, or you could start up either the existing lab or lab script from here if you want to get a shortcut on it. And then we'll go through the slide deck on the graphics, we'll take a break, and then we'll spend the majority of the time going through this graphing exercise. So without further ado, I'm just going to bring back up the prior exercise and refresh a little bit where we left off, just because I want to highlight kind of the functionality and the set of functions that we had stopped on. We were on step 19, I'm actually going to skip this part of 19, which you can, well, we'll just dive in and we'll just do all of 19. I think it's pretty straightforward. And then really this section, creating a new set of vectors and adding those into a data frame is really what I want to highlight, because this is how you could, inside of R, generate your own set of data to work on and do analysis and manipulate. I think that there's two main ways to get your data into R right now. The first being to generate like a spreadsheet in Excel, for example, and save that into a format that you can, be comfortable importing into R. We've covered that through this .csv or comma separated values format. And then this method where you can actually generate these data directly in R itself and then do analysis on those. And so this is kind of where I want to just start off. Let me open up my R session. For the sake of this, if you want to just follow along, you can. But I'll really recommend when we get to the graphics section that you can open up your own session of R and work on that as well alongside. So recall last time that we had this data frame that we had called size, and we just read it in with this read.csv function. And then we were using this file.choose option to just create this file browser that lets us navigate to that data where it's stored on our computer. So I'm assuming now that you were able to download that data and have it, available to you. I can refresh that Google link again in the chat if you need to go and get the data. Then you can do that either now or during the break would be a good time, since you don't really need to download anything. Following along with the slide deck is fine. But recall that we import that data like that, let me just, for a quick refresher, remind us that this is a .csv file. I can open it in Excel and it looks like a regular table and spreadsheet. Each row is a single observation, right? And then we had columns that contain the data about our observations. In this data set, we had a column that told us the common name of each fish that was sampled. And that was categorical data because it was telling us about the names of the fish that were sampled, and different from something that's numerical data that's measured about an attribute of the fish. And if I were to import that, because it's a .csv is the extension here, means I need to use the read.csv function in R to bring that in. And when I look at it here in my R console, I see that it has imported in the same format and structure as I was viewing it there in Excel. I have rows, and then I have those three columns. And I often will look at this structure, str, of my data set, which tells me that I have 15,460 rows or observations of three variables. And then it tells me what those three variables are: common name, length, and weight. And it tells me a little bit about how those data are stored in R. The common name is a character, and you can see that each, these are just kind of random kind of sample of some of the values in that field, and they're in quotations, and that indicates that these are characters in R. relative or different than length, which is an integer, which are whole numbers, or a numeric for weight, which is these decimals. And then let me just open back up to the exercise and review where I was at. Let's review really quickly. This was the t-apply function. This let us look at the mean of the weights of each fish, but instead of just taking the mean, sorry for all the scrolling back up and down here, but I just want to look at the top of this column. Instead of looking at the mean of all of the weights, the t-apply let us group these observations by fish common names, and then look at the mean for each individual type of fish. So in order to apply this function, I had to call and say, I want to look at a statistic on the weight field. So I'm indexing into the size data frame and calling the weight column. And then I tell it that I want to group those weights by common name. And so that's grouping and basically splitting up and saying, I want to create a separate group where I'm just going to split apart. Every time I see a spotted sea trout, I'm going to take its weight and put it in a separate group. And every time I see this striped bass, I'm going to put its weight into another group, and so on and so forth for each different common name. And then I'm going to apply a function to that different set of weights. And in this case, that's a mean. And you can see here how that works. There's a lot of different common names in this data set because there's 15,000 observations. But this function allows us to quickly calculate the means of all of these different types of fish. And we can very easily see, for example, the African pompano weighed quite a bit more on average than, for example, Atlantic crocker. And then I'm going to actually just assign this to a variable or an object in R. So remember this arrow function or assignment operator says I want to take this output and assign it to an object. And now I could just look at this object itself. And that again looks like the same thing I just looked at, but instead of running the whole line of code I just did, I'm only running or looking at that specific object name that I just assigned. So maybe instead of the mean, I want to look at, say, the standard deviation. How variable were the measurements for each type of fish? That would be SD is the base function in R to calculate that. Some fish you'll say have NA returned. That is for where there was only one observation collected or one of those fish sampled. There is no variability in those observations when there's one sampled, so you can't calculate a standard deviation. If you had a 0, that essentially means that every fish sampled had the same weight observed. And then where you have higher values, it essentially means that you on average, saw the mean, but fish could be very much higher or lower than the mean relative to another kind of fish that had a low standard deviation. Usually when you collected sea bream, you got a weight that was very close to the mean. Whereas when you collected a scamp fish, you may see a weight that's farther from the mean, from its respective mean. And we'll see in our graphing session today how we can graphically display this type of variability and understand that visually as well. So I may also want to look at how many observations were collected for each of these fish, and that's the length function. And so you can see what I'm doing here is I'm just running the same tapply command grouping the weights by common name, but I'm changing what function is being written or run on that grouping. So length tells us how many observations were in each of those groups. So yellow snapper, there was 171 rows in the original size data frame that had yellow snapper observations. There was only one swordfish, one spotted hake, and so on and so forth. If we wanted to see all these summary statistics side by side, we could use the cbind, which is just column bind. It's just simply saying, put these observations together in a column format next to each other. And so I'm just going to put these next to each other using the column. And so I can see very clearly information about the mean, standard deviation, and number of samples collected for each fish in that 1,500 or 15,000 row data set. There is a ton of different fish collected, so this is still a fairly large data set, but scrolling down through it, you can see it's probably on the order. I didn't save this to an object, so I can't tell you without like looking more clearly at this, but there's probably 100 different fish sampled. This is summarized from 15,000 observations. And so we can see, African pompano, one fish collected, and it was 4.6 units in weight. There's 224 Atlantic croaker. They're quite a bit smaller in general in weight. And then the standard deviation again tells you on average how far each observation is from its mean. And so it tells you about the variability in those 224 samples. Is there any questions on this part so far? Again, this is all in the exercise notes as well. And so I've tried to type up as clearly as I can. And then I will just highlight again for folks, the exercise notes are in this exercises directory. And then if you go into the scripts, what you'll find is a line by line run through of what we're doing here and what we did in the last session with some comments included on that. And so it's hard and difficult to comment as I'm typing and explaining what everything does. But all of those resources are there for you to review as well. What I want to do now is consider, for example, that I had gone out and collected some additional samples myself. And I want to record those. Because I only collected a handful of samples, I'll just record them directly into R. And then I'm going to combine those with this existing NOAA data set. And so to do that, I'm going to start by creating 3 vectors. So we went over some of the classes of our objects in the last session. Right now we're working with a data frame, and very commonly we'll be working with data frames. That's one of the most common ways to store and work with data in R. Remember that a data frame is 2-dimensional, meaning that it has rows and columns in it. And what's unique about a data frame is that the columns can store different types of data. We kind of saw that in the size data frame, where the common name is a character data set and it's categorical data that describes the name of each type of fish, whereas the weight and the length columns are numerical data and they're measurements about those fish. And so that's the unique property of a data frame, very commonly the types of characteristics or attributes that you want to record about your data will be a combination of categorical and numerical data. What we'll do now is we'll show you how to create a vector. And a vector is one-dimensional, meaning that it's just a string of either numbers or categorical data. And I'll just, without further ado, go through that now. I'm just going to name it. I'm going to start by just naming this and calling this assignment operator. And then C is the function for creating the vector. I don't know why they called it C, it's just C. And then I'm going to put in quotations each of the fish that I sampled. And I'm going to just deviate a little bit from the exercise, and I'm just going to copy and paste some of these fish names because I'm just generating a set of fish names. It's not really important what I obviously didn't actually go out and sample these fish. If you had, it would be really important if you were generating these data on your own that you wanted to match the names of the fish exactly how they were written in the original data set. And so by copying, I'm doing that myself. But if you had typos here, they would actually be recognized by R as a different type of fish. And so R, of course, as we've talked about, is very it's very persnickety about typos in terms of lowercase, uppercase, symbols, spaces, everything like that. And so if we want this type of fish to match exactly and be grouped in to calculate a new average, for example, of Atlantic cod that includes this 37th observation I've just made, then we would need to type it exactly how it appeared in the original data set. So I'll just run that line. We can look at it. Now you can see essentially it's one-dimensional, meaning that when it returned, there's not rows and columns anymore. There's just a list or a set of characters, strings that are returned, and there is 5 values in that. And then I'm going to similarly set some lengths. And again, the lengths here are not very important in terms of what the actual values are. We're just getting a sample of what it takes to record new values in this. So I have no idea if those are reasonable lengths or not for the fish that I just recorded. And then the weights. And I know the weights are kind of like, I mean, I just took the mean of the weights. So you can see they're kind of like between 0.5 and sometimes up to like 10. But I'll just make up some weights. And then just to trick us a little bit, I want to put an NA in for one of those and say, maybe I forgot to measure the weight of one of these fish. So we can look at all of these outputs. Again, these are all vectors that I've just created. And now what I'm going to do is combine these vectors into a data frame. I'm just going to call it new obs. And now the call for a data frame is data.frame. You'll notice sometimes as I code that I put like spaces or extra lines in things. It's not necessary. You could have a very long line here, but often people like to have kind of line breaks. So I'm just going to follow that kind of convention. And what you can see here that I'm doing is here I'm naming what's going to be the column in this data frame. So this is just the name of the new column that I'm generating. And then this is going to define, pulling from this vector, the values that go into that column. And same for length and same for weight. And so that'll, okay, you can see what, sorry, that'll make sense when we look at the output of this. But I got an error. I said differing number of rows. So I can't make a data frame where I had five common names, 6 lengths, and then 5 weights. I accidentally, as I was going, typed too many lengths. So I'm going to delete one length and then rerun that line to overwrite my length vector. And then I'm going to rerun new obs. And now that worked. If you were collecting your own data, of course, you would want to be more careful about just deleting an observation. You would want to make sure that you were captured your data correctly. And it would make a lot more sense when you looked at new obs. And of course, this reflects very similarly the original size object that we were looking at. And here you would spend time to go row by row and double check that all of your data was entered correctly. And that data frame looks good. I could look at the structure of it. Oops. So kind of the same inspection I was doing on the original size data frame. As it is, this says numeric instead of integer. That's not really that concerning. I could force this into an integer format, but for my purposes, that really doesn't matter. And then let's just see what happens if I take the mean of this new obs weight field. Recall that I had an NA in that field, and so I got this NA back. So what can I do to get a weight, mean weight that makes sense? I need to tell this function to ignore the NA and just calculate the mean of the values that are not NA in that. And I do that by saying NA.remove is true, or just remove the NAs and calculate the weight. I could also, just for the sake of complexity, say there's another function here, na.omit. And let me just run this outside of the mean, na.omit, new obs, weight. And this is going to return here just the values that are not NA. And then I could wrap that in mean. And I get the same result. So multiple ways to get to the same answer. And then finally, what would I do if I wanted to combine this with the original size? I'm just going to look at the first 6 observations again. What if I wanted to combine my new obs with this size, original size object? Well, I could just combine them directly, and I would, instead of using cbind, which binds them by columns, I want to bind these by rows, which just means I want to append these new observations down as new rows onto the size data frame. So instead of just doing that directly, I'm going to show us how to add a new column onto each of these. And I'm going to add a column that's going to specify who the observer was, so that after we combine these two data sets together, I can still retain the information of who sampled each fish or record in the data sets. So remember that we can index to a particular column with the $sign into a data frame. And we could also just add a new column by basically indexing to something that doesn't yet exist, like size observer. Oops. And then assigning that to a value. So size observer is not yet a column in this data frame. Yet I'm indexing to it anyways. And what that's going to say is tell R that size observer is a data frame here, or is a column in this data frame. And more so, its value is Noah. So in the original size data frame, I'm going to end up having a column that is named observer and just says Noah for every row. And I'll just look at head size again. And so now you can see what that did. That created a new column here called Noah. And now what I want to do is I want to create that same column in my new object, but I just want to make it my name because I collected those samples. And then finally, I'm just going to combine these two data sets together into one big data set. I'll call that size new. And then remember that I was describing how instead of cbind, which would combine the columns together, I want to combine these as new rows. Basically, I want to take the size data set and I want to just add these as new rows underneath it. And so that's rbind. And I specify the two objects. The nuance to rbind is that you have to have the same names for each of the objects you're binding together, the same number of columns and the same naming for them. But I ensured that when I created these objects, or at least the new obs, that was the case. And then I can look at size new. This is a very big data frame, of course. Because I put new obs second, those observations would be at the very bottom. And I didn't actually put this in the exercise, but this is really a nice secondary function to head, or complementary function to head, is tail. Instead of looking at the first 6 observations, I could look at the last 6 observations. Oops, I just looked at size. I want size new. And I see the 5 I added after the last Noah observation. And then finally, I can write this out to a new file if I want to. Instead of read.csv, I'm going to write it to a CSV. And so what I'm writing out is size new. And then I have to tell it the file path that I want to save it to. I'll just save it in my data folder. And so what I'm going to do is I'm going to right click on data, and then I'm on a Mac, so I'm going to hold down Option to select the file path. And I can help you guys get file paths if you want. And then in quotations, I'm going to paste that and then just followed by a forward slash size. Not going to have any um, I'm not going to have any fun with the naming of this one. I'm just going to call it size new. And then I have to put the .csv to tell it I'm going to name that as a CSV. And running that function now just created this data set in my output directory here. I could open it. I could tell R that I don't want this column here. It's pretty easy to say I don't want the row names in my output data set. For now, I'm not going to worry about it. But you can see we have the original size observations. We've added the NOAA column or the observer column. If I scroll all the way down, you'll see that I have my 5 added at the end there as well. So I'm going to close that. And to recap just the end of this exercise, we were able to, we were able to use tapply to group things by different common names and apply these multiple functions. And that allows very quick and easy statistical calculations across different groupings. But the main part that we were able to accomplish here was generating some quick data frames within R, manipulating a little bit about those, including adding columns, and combining data frames with this rbind function, and then writing out a new writing out a data set from R back onto our disk. So now we can both import data, manipulate it in R, and export it back into a file on our computer. Any questions with that? I'm going to remind folks again that I put a set of practice questions at the end of last week's exercise that uses this Washington fires data set. I'll look at this chat question in just a moment. These should be addressable with all of the tools that were introduced with this exercise 3 concepts. And so I really encourage, just for practice, to come in and try to answer some of these. And I'm happy to, I didn't put answers in here. If people do respond to me, I can work up a short script that would answer these and provide that as well. But these concepts, often require a little bit of practice to really grasp and retain. And then the question about file paths. I'm looking at the comment. in the chat, and it almost looks like, is it that there's two sets of quotations on the first? Okay, yeah, file looks like a decent answer. Dave, if this answer from Nathan uh, doesn't address it, then maybe we can address it either at the break or at the end of the session, because I think it does sound like a file path issue. And these forward and backslashes are definitely different between Macs and Windows. And it also may be an issue with the quotes you have, but I would need to look, I think, more at like directly at the code you have, than just the error, maybe to understand exactly what's going wrong there. Okay, okay, try. I'll respond here and do a try. All right, I'm gonna see how that pans out as I open this slides for the graphics and check on my attended schedule here. Okay. All right, so maybe that was a little tedious. I think that you'll find is a good little bit of refresher. There will be a little bit more refreshers of the exploring data activity today. Because what you'll find is as we go through this graphing exercise, to really investigate and understand our data, kind of broken down into different components, it's really helpful to say summarize it in different ways or subset it in different ways and then display graphics for those different components. And so you don't really need to have a strong understanding of last week's material for today. Uh, that said, we will be using some of the functions and introducing just a couple new ones, at least one new one. Um, uh, and so you'll be a little bit more comfortable with that review, and hopefully things kind of progress from here. So we'll just start with a quick review of data types, because I think that's a nice concept to just get some review of overall. We'll look at how the different types of graphs that we can produce kind of will kind of tie into those data types. And then moving into the exercise, we'll really look towards getting comfortable coding these graphics up in R. I put this chart here as kind of trying to reference like these data types. The big difference I want to call out is this categorical versus numerical is kind of this top level hierarchy. And I have some examples to follow here. When we think about this, really we can think about kind of that size data set, the fish length data set, where categorical data often looks like words. Very often, it's not always. We could code things numerically. We could say males are zeros or females are ones or something like in our data set, and just have numbers kind of relating to a set of categories. But very often these are words, or they're collected as words, and they're known generally as words, but they may not be stored that way under the hood. We think about them as characteristics of objects or things that we want to measure. So what color hair do you have? What color are your eyes? Um, uh, what species plant are you looking at? That type of thing. And very often things that you would count. So when I think about counts is if you looked into an area, say, how many elk are here, right? Really, that's you're looking at characteristics of species, but you're counting something. And then depending on whether you can order those categories or not, puts you into this ordinal versus nominal dichotomy under categories. So ordinal data would be categories that could be ordered, like I disagree, or I don't disagree, or I uh, I was looking up the scale last night. It was strongly disagree, mostly disagree, neutral, agree, strongly agree, right? Those are categories, but they have some order to them. You don't necessarily know how much change there is going from a strongly disagree to a disagree category, but you can order them in that kind of hierarchy. In nominal data, there's no order. So if we're thinking about like species names, if there's elk, coyote, wolves, owls and whatnot, there's no order to those. At least in terms of my non-wildlife brain. If you're, really a wildlife person and you're thinking in terms of a trophic food web hierarchy, maybe you are posing order in terms of that food web. But so these aren't strict, like I'm going to stumble over my words here a little bit, but I'm going to say, just because I'm suggesting like, oh, species names are always nominal, that's probably not the strict case. You might be able to make an argument where, hey, I'm using these species names as proxies for their position in a food web. And really, I'm thinking about not really the species names. Really, I could reclassify these as like herbivores, carnivores, mesocarnivores, or something. And that would be a better category to use for those. And that's really that now that becomes ordinal, right? Because I can order those in terms of that positional hierarchy in a food web. I really hope that I'm not being too confusing there. We'll see a little bit about how these can relate to types of graphics you can produce. But for a moment, let's jump over to numerical data, which are probably a lot easier to recognize. Well, maybe easier to recognize. I don't know. At first glance, they're always going to look like numbers. And I want you to think about measurements, and I want you to think about a scale with units. And so we could think about the length of something in meters, or I've weighed something in kilograms, or I've measured the temperature in Celsius, right? So I'm measuring something, and that scale that I've measured it on has units. Now, there is this complicating factor underneath here about interval or ratio scale. I don't want to get too deep into these, and I've put a link here for you to read a little bit more about. But this has to do with whether or not there's a true zero on that scale. And the kind of canonical example for this interval scale without a true zero is temperature measured in Fahrenheit or in Celsius, where you can have negative temperatures and they're not really, they represent true meaningful values. Whereas in a ratio scale, if you're measuring the height of a person, you could not have a negative value. If you were measuring the weight of an organism, you couldn't have a negative value. So the zero is a true meaningful 0 point. And I don't want to get too caught up in these distinctions. I just want to introduce that concept, leave some references there for you to understand, but really linger more importantly on the distinction between categorical and numerical data. So here's some examples of these nominal data. We've gone out, we've collected some organisms, so we're I'm not a biologist, so I just kind of making up, some things here. But, we're going out and we're doing a fish survey. We're in a river or a stream, we're collecting fish, and we're measuring, what the species of that fish is, what its sex is, and maybe what life cycle stage is. And so when we go to record that data, right, we're going to write down a word for those things. What's the species? It's a sculpin or a salmon or a bull trout, right? What's the life cycle stage? I don't even, a fry or a red or other ones I don't remember. And so you can think here, you're writing down words, right? Ordinal data, again, I went through this opinion one. If you were measuring water temperature just with your hand, and you just were like, hey, I've got to really, I've kind of calibrated my hand over time to measuring the water temperature. And I've been collecting these fish as I go in there. I just say, is it cold, lukewarm, or is it hot in the water? And I haven't measured that with the scale. And I'm going to write down a word, right? So that kind of indicates to me that it's categorical data, but I can order these in some hierarchical order. And so that's telling me now that it's ordinal. And so I like cold, lukewarm, hot. And so I may be able to do something with that a little bit different than if it were just nominal data. Interval data, if it was temperature, I don't know if I put more to come on why. I think I did put another slide just for like you to read on that really, but I just described all of the stuff already that I wanted to say. And then the same with the ratio about how height, weight, and size. Really, you can't have negative diameter of a tree. It's not a meaningful measurement. And so those indicate to you that 0 is a true 0 point and it falls in a ratio scale. I'm not going to cover this. This is just what I was kind of talking about the temperature being on the interval scale. As long as it's measured in things like units like Celsius or Fahrenheit, you can have negative values that are truly meaningful. And they limit how you can divide or multiply. And we're not going to deal with any of that in our examples of our data analysis today. I just wanted to have this slide in here kind of for posterity's sake for you to refer back to if you want. And I just had somebody note in the slide here, that's a really good point, that life cycle stage probably should be under ordinal because you are kind of sequentially growing up or developing through those stages. That was my my non-biology thought in there, but that's a really good point. I will fix that slide after this work. So I put these as just kind of questions in here that you can come back to and think about. But these are the kind of questions you might see on like a intro, statistics quiz. Basically, like, hey, if you want to, come back and refresh or um, quiz yourself on some of these. The hardest one I put on this was this number 4. So I'll just review that one. And then I think these other ones that you could probably answer just by referencing those prior couple slides. But this Mohs scale is pretty interesting. This is just what scratches what. So mineral hardness is ranked based on this scale. And if you scratch something, then you get a ranking. But if something else scratches you, then it's harder than you. And it's ranked 1 through 10, but it's not actually units. It's just a ordinal scale telling you that your diamond is harder than your steel, for example. And so that's kind of an interesting one where it looks like numbers, but actually is a categorical scale that is ordinal. Here are some types of graphics that we might produce with these different data sets or different data types. So if you're working with nominal data, you might think about the rate at which something occurs over a period of time or within a data set. Or similarly, you could look at the proportion, which is really that frequency divided by the total number of events. These are really kind of the rate over a period of time or within a data set. This is species diversity created into a pie chart. And so this is nominal data because each observation is classified or categorized into a different type of species. And then out of all of the observations, I don't have a count of how many total observations were here. What you can see is most of them were insects. So while this isn't directly a proportion, excuse me, you can fairly easily read off of a pie chart like this that over 50% of the species on Earth are insects. And then after that, the next highest proportion is vascular plants. And it's kind of hard to read exactly what slice of the pie that is, but that may be what, 20% or a little bit less, and then so on and so forth. This second chart, this was species identities assigned to products from takeaways and fishmongers. So the bars are divided according to retail origin and global IUCN red list conservation status of each species highlighted. So I didn't read a great background on exactly what all of this data generated was, but essentially you can see that what they have recorded here is species names. So that indicates a categorical data set. And it's not, there is no inherent order to these species names. So it's nominal data. And then we're displaying here the number of samples. So how many samples were recorded? Most of the samples were of the spiny dogfish. followed by the starry smoothhound. And then this is called a stacked bar plot. It says within this set of samples, how many of them were fishmonger samples versus takeaway samples. And you can see, for example, spiny dogfish was dominated by takeaway samples, while starry smoothhound was dominated by fishmonger samples. And here's some examples of ordinal data. So this was conservation status of EU protected habitats in Ireland. Again, I don't have a great background on these data sets, but bad, inadequate, and favorable, right? There's obviously some inherent order to these categories. Again, I guess I could start that they're categories: bad, favorable, inadequate, right? They start out as categories, and then we could order them. Probably bad is the worst. Inadequate is probably, it's obviously not good, but it's probably not as bad is, and favorable being the best. And I'd have to read a little bit more into understanding exactly the nuance of difference between those categories, but there's clearly some order to them. And so that is ordinal data. And now we've also classified this according to the frequency of observations. There's a little bit more data set that we could, or like summary statistics that we could incorporate for ordinal data if we wanted. I didn't go too far down that path, but I did include this kind of a stacked bar chart again of this kind of scale where you say not important, little important, moderately important, very important. And this was a survey of I can't remember what this survey was about, but it was something ecological, and you can see the responses having to do with restoring to a historical state, restoring ecosystem function, planting native species, increasing biodiversity, human use values. So people were surveyed according to these values, and they rated them on this scale. And then the stacked bar chart shows, relative to this scale, what respondents were most concerned about. And so if you look, say, for example, very important being green, respondents were overall, most concerned about planting native species and habitat for native wildlife. And if you look at where I say maybe green is a very small proportion of things, or your brown and your tan are high proportions of things, restoring to historical state, erosion mitigation, climate change reduction, aesthetic value were like of lesser importance overall. I don't know where that survey data came from, or if it's even a real, in this case. I didn't have a citation here. It was just a image that I found. So I don't know if it's even real data, but I thought that it was an interesting example of displaying that kind of ordinal data in the stacked bar chart way. I think there's what we'll see is a few more ways that we can look at continuous data. As you'll see this, because it can use most different methods of data analysis. We have our box plots and our histograms. Or I didn't show I kind of didn't get all of the examples I think I wanted to here. We'll see in the exercise two variable plots that include scatter plots and line graphs. Here we're looking at single variable plots. that are box plots and histograms. So we'll see kind of some more examples in the exercise. But here we're looking at what I was describing about the spread of distributions. So you can see where the majority of your observations are located, as well as something about the variability or the spread in those observations. And in here, they were looking at the length of fish caught across different locations and different depths. And on the right side of this, it was fire sizes at present and predicted into the future. And this is a histogram. And so it's showing on the x-axis bins of fire sizes, and on the y-axis frequencies. And so here you can see a bin saying, I don't know exactly the tick marks here, but this might be 0 to 50 acres. And most of the fires of the present are in that lowest bin. And there's very few fires in this high bin. And the distribution kind of looks like it's kind of generally decreasing as you move towards higher and higher bins. The analysis here shows the interpreting these two distributions. What happens is, if you remember kind of the phrase, at the height of COVID was flatten the curve. And for COVID, that was, a good thing because we didn't want these spikes in infections occurring. Here, flatten the curve has kind of been a bad thing because it suggests in the future, actually what's going to happen is there's not going to be so many of the fires being such small sizes. We're actually going to move those fires into these higher bin sizes so that our fire distribution actually looks like it's flatter, where more of the observations are occurring at higher size fires. And so in this case, we've actually moved from having fires that are very much often smaller in size, shifting those smaller ones to being larger is kind of the main takeaway of that figure. Okay, just a couple kind of housekeeping items here, and then we'll take a break. I'm going to go over a lot of our plotting in kind of the base graphics and this ggplot function. The base graphics is like really convenient. I often use the base R graphing myself. Doesn't require any extra plots to be installed. It's really quick and straightforward. It is a little bit tedious understanding across different plotting options, all of the arguments and ways that you need to finesse your plots. And this ggplot function is generally a little bit more straightforward, and it has this unified interface where the options are much more streamlined across different types of plots. And so we'll see examples in both of these functions. ggplot's also really convenient because you get this access to these different themes where you can quickly and easily adjust kind of the visual of your plots. And you can install additional packages that have even more themes than the base ggplot package has. And then I've just included this with a reference for you, but it has this kind of layered grammar of graphics. And we'll see a little bit of this as we go, where many of the different types of plots that we have are kind of built up by the same layering of code. And so I've referenced this for you here to read a little bit more about that. And then I've referenced a couple of graphing tutorials as well here that you can look into. And then with that, I guess I'll just pause for questions, and then we can take a break and come back for our exercise. Okay, let's break till 12. Oh, my math is even hard to do in my head right now. Um, let's just go to 12:45, just over 15 minutes. So quarter till whatever time zone you're in. I'll hang around a little bit for any questions, and then I'll probably take a little break myself. And then when we come back, we'll just run through the graphics exercise and wrap up.
Cool. Sounds good. Thank you.
Great. Oh, I see.
Hi, Colton. Yeah, thanks.
Yeah.
Thanks so much. It's a great session. I missed the first one, but I'm catching on pretty quick here. Thank you. All of the details in the Google folder are great. Thank you. I wondered, just by chance, will the recordings be available though? That would be nice to be able to look at them later. Or I mean, it's not essential because everything in the folder is so great, but just wondered. Mm-hmm.
Yeah, Laurel, I saw you just turn on your camera. Do you want to address that?
Yeah.
Also, Laurel, if you send me something, I'll post that in the folder as well. So as people download that, maybe that they can, have that for future reference as well.
Okay. Yeah. So we are making all the recordings available for each of these sessions on the Native American Fish and Wildlife Society website under the membership portal. So
Okay.
utilizing your member info login, and then you'll be able to access this folder along with all the other folders related to all the other information we have for members as resources related to fish and wildlife management. So there's a lot of other information out there related to other offerings where we have beyond just this art course. And if you have any difficulties accessing that, you can feel free to get in touch with any of us here at the society. And we have a membership coordinator, Heidi McCann, who can also help you get back into your membership login information if you don't have that. And yeah, we're all available to help.
Okay, great. Thank you for that. Thanks.
Colton, can I stop you for a second? And I see you're talking, but we're having problems hearing you. Can you check your settings?
Okay, give me a second.
All right, I heard you right there.
Try to put my headphones back in. How about now?
Yes.
Oh man. Well, let me see if I can get them switched over. How about now? Yeah, okay.
Yeah.
Wow, I had such a nice speech going there. I was in the zone. Glad somebody spoke up though. Um, Basically, I was just saying we have an hour about to go through this last exercise. It'll be a combination of fun and tedious, as we've all come to understand that data analysis in R is. But I think that you may find this session to be a little bit more gratifying in terms of the instant result that you get from visualizing your data and how creating clever and unique visuals to complement your data analysis is not just a fun process and helps you understand patterns and processes, but also guides your story and your ability to communicate your results to others. And so I think that this session in particular will show you how that how all of kind of what we did last week can kind of come together in terms of the ability to produce content in a creative way that you can share and display for others. So that was the quick gist of what I did, but maybe less um, less clever my second time through. I don't know, but I've just opened RStudio. I've gone ahead and gone to File and started a new R script here. I forgot to add that in the exercise, but hopefully you can refer back to the previous one to go ahead and do that. We're going to use a new package this time called ggplot. The actual package that we're going to install is called ggplot2. And remember that a lot of functionality in R is available in the base R, which is just the commands and functions that come along with when you originally install R. But because R is open source, there's a whole suite of additional software packages that you can download and install for free. And these simply provide new functions and further functionalities that you can take advantage of when you're doing your data analysis. ggplot2 is a very common plotting framework, and I personally use it kind of interchangeably with the base R plotting. So what I'll show is kind of some things we'll cover both in ggplot and base R, and just kind of give you the options to use whichever you desire. And I will just highlight from the start that just like last week, this is really an introduction to what's possible. And the layers and levels of what you can do and where you can go with both plot, the base R and ggplot functionalities goes far beyond what we can introduce in the next hour. So without further ado, I'll just dive in, but just recognize I've provided resources probably a little bit scattered, but throughout the exercise, the slide deck, and then I will just highlight also, I added a couple in this readings. Don't get too thrown off, I guess, by calling it readings. There's like these cheat sheets, or like base R graphics cheat sheet. They kind of tell you like, oh, I want to create a bar plot, I'm going to use this, and then here's like some of the arguments you might add, and then there's a ggplot2 cheat sheet that you'll see. If you review this, you'll be able to see like, oh yeah, we added or we used like, you know, geom point or geom line on geom line is here on our exercise. So without further ado, let's install ggplot2. Very simple. It's called install packages. And then in quotations, I'm going to say ggplot2. We have to just make sure that we match the case and spelling of that package. And then I'm going to use command return to run that line of code. If you're on Windows, again, it might be control and enter. And that installs the package, but doesn't load it. And so I've included some of this detail in the exercise notes, but you only need to install the package once. This downloads it from the internet into your R folders. But when you actually want to use it for your code, you need to load it. ggplot2 load. Oh, sorry, not load, library. Just trying to use a name of something that I was describing in verbally, but library is the name of the function to load the package. And so you only really need to install it once. If you move to a new computer that you haven't used R4 on before and you want to use ggplot, you'll have to do this install.packages. But every time you open a new R session, you have to run library ggplot if you want to use that that functionality. And that just says I actually want to basically like turn on that extension in this session of R. Okay, so we're gonna just go ahead and load the fish data again. So we've gone through this method for just loading the fish data. And I'm gonna navigate where I've saved it in my mess of files. I'm just loaded the original fish length data set, not the new one that we saved out earlier. And maybe I'll look at it just to make sure everything got imported correctly. I think we covered this in pretty good detail, what each of these data types was for this. Again, this is categorical, nominal for a common name, and then numerical. Now we could say this is numerical ratio data for both weight and length. The first thing I want to do is create a bar plot. So if we just, I'll just flip back to the slide deck really quick. We look back at our nominal data example here. Two really common types of charts to create for this kind of nominal data would be a bar plot and a pie chart. And so we'll just start with creating the bar plot. And we're going to create a bar plot that shows kind of similar to this one, the common name on the x-axis here, and then a bar that goes up to count the number of samples in our data set. So if you'll remember from last week, we did a little bit of this activity where we created a distribution of the counts using this table function. So here, I just named this fish instead of size. Don't be thrown off by that. It's just whatever you name the object here is what it's being called in R. Whatever we loaded with this command is what actually is this object name is pointing to. So while now I called it fish in this exercise instead of size, it's pointing to the same data set that we were working with last week. Now I want to make a bar plot that shows on the x-axis the common names, on the y-axis the counts. So what I could do is I could just start by creating a table of the counts by species name. So just like this, I'm going to call table, and I'm going to index within the fish data frame the common name column. And remember that all of this command is going to do is it's going to essentially group all of the observations by common name and just count how many there are for each group. So this is just that output. Okay, I want to assign this to an object so that I can easily work with that. Fish dist. And then while this is in a table format right now, I actually want to coerce this into a data frame. Oops, I didn't put anything in there. Okay. So now you can see what I've done is I've taken that table and I've just made it into a data frame. And then we went over this in the last exercise as well. If I just want to order this from largest to smallest, essentially what I'm doing is I'm just indexing this data frame. And in my index of the rows, I'm just specifying the order for them to go. So here I'm indexing that data frame. And then, oops, I'm sorry, I need to call order. And I'm just indexing it to say, I want to order it by the frequency column. And because I want to go from largest to smallest, I need to say decreasing equals true. And then remember that because this is a data frame, I need to put a comma and index it with two like two elements in this square brackets. But I don't want to subset the columns at all. I just want to return both columns. So I'm not going to put anything after the comma. I'm just going to leave it blank and run that. Again, I'll just say it again. This code is commented in this script, graphics in R, that you have access to on the Google Drive. So while I'm not commenting things as I go, because it's really hard to describe code and comment all at the same time, you'll find it commented here and a little bit more spelled out in the exercise as well. But I'll just look at that, and now I can see that what I've done is I've just taken that data frame of observations, and now the frequencies start at the maximum value, and they go in decreasing order down to the minimum value. Now for this bar plot, I messed around with this a little bit myself before generating this exercise. And there's just so many names here that it got really busy. You can imagine a bar plot, you would need a lot of wide space to place all of these columns. And so for the sake of simplicity here, I'm just going to plot those observations that have 100 or more counts in them. So how would we do that? Well, we need to index this data frame again. And now we want to subset it using this frequency column to only those rows where that frequency column is 100 or more. So again, I said we would rehash some of the concepts from last week. Here we are. I'm going to index this again. And now I'm going to go into the frequency column. And I'm going to compare each value and say, keep it if it's greater than or equal to 100. And I'm again going to leave the column elements blank, so I return all the columns. And essentially, before I look at this, what I should expect, first of all, I'm always doing two things when I code anything here, is I'm thinking about what I should expect based on what I intended to generate, not actually what I typed in, but what I intended to code. And then I'm looking at the output and making sure that it matches those expectations. So what I expect this object to look like now is what I just pasted out, except it's missing all of these rows where you had less than 100 observations made. And so I can look at it again and just confirm now. In fact, it starts at 112 and it goes up to the max number of observations. Maybe you don't like these column names because they're a little meaningless. var1 is just by default what happened in our process of creating this table and data frame. So we could rename these column names, and we would do that with the call names function. I should just, I didn't do this in the exercise, but we could just run this, and this would return for us what the column names of that object were. But if we create an assignment operator and we pass to it a vector, we could rewrite over and specify what we want these to be. And so I'm going to say I want it to be common name. And then in quotations, I'm putting that in quotations because it's now a string there, and you notice these are both in quotations. So common name in quotations, and then comma space, and I'm just going to say count. Frequency would be fine. I'm just going to call it count. And now when we look at this, you'll notice the top here, the column names have been changed. So really easily in R, we can generate a bar plot with this object. We want the bar plot to be the counts. but then we want to specify what the names on the x-axis are going to be, and that's the names argument. And that's going to be the common names. I'm just going to open up my bar, my plot a little bit more so you can see it. That's pretty decent. It's a start. But you can obviously see that there's only a few names specified here, but there's far more bars than there are names. So one thing that we need to do is we need to rotate the names on the x-axis. So I'm just going to copy and paste that whole command. And then I'm going to add LAS equals 2, and that's going to rotate those names for me. You might have to make it a little bigger. I'm sorry if I'm cutting things off now, but I can still see that there's not enough names per each column or bar. And my problem now is that I can't read the names that are being cut off down here. So this par is defining the parameters of this plot. And I want to specify the margins. And this goes bottom, left, top, right. It's a little confusing. You'll see it in action in a little bit. I'm passing it a vector and saying I want a large margin on the bottom, decent one on the right, and it can be pretty small on the top, or sorry, a decent one on the left. It can be pretty small on the top and on the right. And then if I just copy and paste the same bar plot command again, I can run it, and now everything displays in my window. I can visually just check that I have one name for every bar. I have them ordered from largest to smallest. ggplot is pretty convenient, but you have to get a little bit used to the way that it is set up. So the way that you have to set up your ggplot is you specify the data, the data set that you want to map on. And then you have to pass this AES function. And this is called aesthetics. And essentially it describes how you're mapping, the columns to the visual properties of the next line that we'll specify, which tells it what type of plot to make. So we say X equals common name and Y equals count. And then I'm just going to put a plus because I'm not quite done yet. You'll see this is the way ggplot functions, is you have to specify this first thing, which is like, hey, I'm going to plot stuff from this data set with these mappings of the columns, and I want to make a bar plot, which you can do by this geom call, _call. All right, similar to above, we've created an initial bar plot, but we have some cleaning up to do. I had to Google this online, so just bear with me as I type this out. Theme addresses like elements of the axes that we want to change, and it's clear that we want to change some stuff about the x-axis text. because while it does display all of the names of the fish, they're running way over each other. So axis axis.text.x, and then element underscore text. And we want to say the angle is 90. So that'll rotate them. We could just look at that if we want. Not bad, right? Maybe you don't like how they're all centered there. So V just oh, that just centers them that way. We wanted H just. You'll see now they're all justified horizontally to the right, up to the side of that tick mark. Now, I can tell that there's a lot of stuff I did in this exercise. And for the sake of just making it through here, I want to just skip some elements that I've written in here. But I did go on to clean this up a little bit more, essentially by changing the theme here and the x-axis label. But I'm just going to skip over to the pie chart because I think it's a good time for that. So let's consider, oops, I didn't mean to save that. Let's consider making a pie chart of fish lengths. So I'm going to start by just looking at what the fish lengths look like. For this pie chart, we're just going to create an ordinal variable from our fish lengths. Instead of mapping, instead of plotting like the actual measurements, we're going to plot fish lengths in terms of short, medium, and long. So imagine if by some convention in your program or your study, that fish lengths of under 300 is short and over 800 is long. So we just need to create a column in our data and then populate that column with short, medium, or long, depending on what the length of that individual fish is. And then we'll use that to create our pie chart. We can see from the summary that there are fish that would be under 300 and fish over 800, and there's fish in between. So we're going to have fish in all three categories. We just covered how to add a category or add a column. I'm going to call it length underscore cat, like this is the length categorized. And I'm going to start by just making it equal to NA. It's not very exciting, but it's added a column in our data. And then I can go in and I can index observations by the length field. So remember, if I just index like this will return all the observations where the fish length was under 300. So if you scope through this length column, you'll see that they're all under 300. So what I could do is I could just put a $sign at the end and go back into this length category field I just made and say that those are all short. Now, if I just look at fish, I'll see that for ones where length is under 300, I have short, and all of the other ones, I still have my NAs. So now I need to do more indexing. So if length is greater than 800, I want to make length category equal to long. And then finally, I want to make one that says if the length is greater than or equal to 300 and less than or equal to 800, you're going to be medium. You can look at the full data set and just scan through and see that your criteria look like they've been applied correctly. You could create a table of that and see how many are in each category. And then we could assign this table to an object and then make it into a data frame. Right. Again, we have kind of the ugly column names. Length, category, and count. And now we have the summary of our data showing fish lengths in an ordinal format with their counts. We could make a pie chart. Our pie chart is going to use the counts, but the labels of the pie chart are going to use the length category from there. Maybe it's not the most interesting of pie charts, but it's very easy to see quickly at a glance if these were true categories, meaningful categories for your data, that, hey, almost 75% of the fish that you collected or observed were medium in length. Almost, about 25% of them were short, and there's a small sliver of them were in that long category. And, you could display that or calculate that in tabular format, but it's very easily conveyed visually as well. I'm going to skip the ggplot example of this. It's in the exercise, but it's just it's a little complex how I had to code it together to get the axes working. So I'll just leave you to revisit that in step 15 if you want. So let's do some box plots next, because this is moving. Oops. We've done, a couple graphs on categorical data. So let's move to some single variable numerical data. And that'll be box plots and histograms at our start. So a basic box plot is really easy. The command being box plot. And we can just look at a box plot of the fish length. This allows you to see where most of the observations are in your data, kind of being around 300 units in length. And then some of the outliers being some, number of observations that did make it up to 1,500, but very, very few. So now you're looking at the spread or variation in your data. If we wanted a ggplot version of this, it's very simple here. We say we want to use the fish data set to plot with. In an AES, we're not plotting anything on the x-axis, right? We're just plotting on the y-axis the distribution of lengths. And then we have to tell ggplot that we want to make a geom box plot. I won't go into doing a lot of dressing up of things right now of this, but we'll at least do one where we dress up a ggplot a little bit more than, you know, leaving this X here like this or like so on. So we're gonna make take advantage of some of the functionalities in the next. Oh, look at that! I misspelled length. In the next spelled it differently here than I did here. Oh well. See, I can be prone to typos here. In this next set of graphics, we're going to take advantage of a pretty cool functionality where we can very quickly generate separate box plots for each different type of fish in our fish data set. So we already have seen through the tapply functions that we actually covered at the very beginning of today's session, kind of going back to last week's exercise, that these fish generally have different average sizes and different variations in those sizes. But it's not so simple to capture or understand that variability just from looking at numbers. It can be much more intuitive to visualize it graphically. And so there's some very nice functionality of doing that in R. But to simplify things a little bit, I want to just subset a data set to only those common fish and then plot those box plots for them so that we're not overwhelmed by box plots. And so you'll recall that we have our fish dist object right now that essentially lists out the commonly sampled fish. And we have our fish object, which has samples for all of the fish. So what we're going to do is essentially index into this fish object and use this fish dist to essentially index and keep only the observations in this fish a data frame where the common name matches one of those commonly sampled fish. I know I'm using commonly too many ways, but where the species name here in the data frame is a commonly sampled fish, meaning that there were at least 100 observations of it in the data frame. So I'm just going to start by naming a data set, fish common, and we're going to index fish to get there. And we're indexing fish based on the common name. And if we do this %in% operator, that basically is going to check whether this fish name is in the second thing that we pass it. So the second thing that we pass it should just be the fish dist list of common names. So here we've indexed fish dist to provide it the 26 most commonly sampled names. And now we're using that list of 26 most commonly sampled names as an indexing criteria on the original fish data frame. And the comma space, again, because I'm going to return all the columns of that. And I can look at this. Let me look at the dimensions of it. It's probably not that interesting to look at now, but it's gone from about 15,000 samples down to 1,300 samples. I should have done this in the exercise, but if we look at the unique fish common names, there was 137 different types of fish in the full sample. And if we look at unique fish common names, I'm sorry I used common so many times here. There's only 26 different types of fish in this fish common data frame. So while we didn't drop too many samples overall, we did drop a lot of different types of fish that were very seldom observed. Okay. There's a little bit of mouth chewing to get here, but now we're going to work with fish common, and we're just going to make a box plot of fish common. Sorry, we still want to make the box plot of what were we working with? Length. We want to make it as a function of the common name. I can type that out. And then I'm going to add this var width equals true, which just means I'm going to vary the width of the box plot based on the number of samples that went into each observation or each common name, how many samples were taken. And then remember about this LAS equals to rotate the axis labels. All right. We're pretty close. I think I just want to get rid of this x-axis label, and I want to change this y-axis label. So when we come back here, make this all visible. X label, I'm just going to put empty quotations. That'll just get rid of it. Y label, I'm going to say is length. I'll rerun that. And then let me expand this so you can see all of the fish names. Oops, just hit saving. So now you can see in this graphic, the box plots that are up high are generally bigger fish, like longer, because we're just looking at length, than the box plots that are down low. The kind of like overall size of the box plot relative to this one that's pretty tall, like this one's really tall relative to one that's pretty short, tells you about the spread in the data. So little Tunny had most of its observations were about 500 to 600 units in length. But some fish measured up to 800 or down to 300 or so units. Whereas something like the northern puffer, it was very unlikely that you would get a fish that varied from its mean by very much at all. Most of its fish were about a little over 200, and nothing was more than this 200 units away from that value. And so this tells you about that variability in those distributions. And then the width of the box plots shows you can see black sea bass was very commonly sampled relative to bluefish because it has a wider box plot. So there's a lot of information that's available for you here. You can look at the overlap of these box plots and see that the king mackerel were always bigger or longer than the Gulf kingfish. But some of the shorter king mackerel we're as long as the longer little tummy, right? Not usual. That's not usually the case because the bulk of the distributions aren't overlapping, just these whiskers on the box plots. But there is potential for overlap between those two fish. Okay, let's see. I am just looking through, and I just want to quickly do some histograms. So histograms are these frequency distributions. They're very simple to set up. They are prone to the interpretation of them is highly dependent on the number of breaks that you have along the x-axis and the position of those breaks. So let's just really quickly look at the number of breaks. So if I just add an argument to say, I want only 4 breaks, whereas this one has, looks like about 10 different classes, or maybe, I mean, I guess if you go all the way out to these ones, more, but if I just want 4 groups, you'll see, hey, that's 4 groups. Now it looks like most all of my fish are very small, right? And I have very few large ones. Whereas here it looks like, well, actually, I have some small ones, a lot of medium length fish, and then very few large ones. But I could also set my breaks out to 40. And now I get a very localized distribution. And so I don't want to go too deep into the histograms here, other than just to highlight how much the number of breaks is, how much your interpretation of the pattern of that distribution depends on the number and the location of those breaks. Instead of passing a value here that's the number of breaks, you could pass it a vector of numbers that tells it where on the x-axis you want those breaks of different values to be. Let's see. I just want to go into the 2 variable stuff because I thought this was pretty fun. So 2 variables being like a scatter plot or a scatter line, line graph. So now we'll use this plot command, but let's look at fish length versus fish weight. You can very easily just look at that plot. You'll see it looks like there's a pretty strong relationship that as fish get longer, they also get heavier, right? So these are kind of positively increasing. If you were to draw a line here, there's a relationship there. I'll go into the ggplot for this example because it has some nice functionality for making these transparent as well. So I'm just specifying that I want to plot on the fish data frame. I want my x-axis to plot on length. I want my y-axis to be weight. And then I want to plot a geom point, meaning I just want to plot instead of like we did a bar plot, now I want points. All right, that's a really nice start. However, just like the BLOS plot, you can see there's a ton of overlapping points here, especially down low at lower lengths. So what we could do is we could add this alpha, equals And we're going to pick a number between 0 and 1. Alpha is transparency. So maybe I want even a lower value here. And maybe you don't like these grid lines and it's a little interfering because gray, transparent gray on a gray background is just, difficult to interpret. So now we can go back to how ggplot has all of these available themes to us. And so theme classic, I like because I'm a basic plotter, just is going to make black and white plot background. And we'll just look at how that looks. Right, so now we're not distracted by that plotting grid. While sometimes it's useful for kind of locating where things are here, it wasn't really helpful in that regard. And now I think this might be the final thing I do unless we want to continue a little bit further, but I just sense that I might leave the next section that went into the fires data set for practice. But I think that there was some really cool stuff I did there too. But here, ggplot has a really cool functionality where if you'll take note of the scatter plot that we're looking at, one thing that you might notice based on this transparency here is it almost looks like there's like different lines or different like kind of trend lines, right? In those observations. I'm kind of looking like right through here, there's like a dark line here and then another dark line right above it. And I kind of wonder, are the relationship between length and weight varying by species? And I think that's a pretty, pretty kind of basic question that we might want to ask about this data set. Because we already looked at, oh gosh, let me look it up. Because looking at these fish was just too much fun. We already saw these two guys. And I just can't believe in my mind that, the relationship between length and weight of these fish would be the same for such body types as different as those are there. And so in ggplot, we can add one more command onto here called facet wrap. And inside here, we're just going to specify what we want to break out these plots by. This is similar to like our groupings with tapply, where we say instead of plotting this relationship all at once for all of our data, we want to break our data into different groups. And for this, we want that grouping to be the common name variable within fish. And we want to plot this relationship for, I don't remember all the fish name, but we want to plot this relationship for each individual common name fish separately. And then we can see how different this curve might be based on those fish types. The way we would do this is we put this little tilde, which is kind of that function symbol we used previously in the box plot example. But because we've already specified our X and our Y in ggplot, we don't need to do that in the function. We just need to tell it what to break them up by. We want to break them up by common name. And then we just run that command. I hope it works. let's see, maybe is the space there not going to be good? Oh, you know what I'm doing wrong? I want fish common because there is way too many fish in the that full fish data set to use. So now we're just looking at those 26 common species. And what you can see is that there are very tight relationships between weight and length for each fish, right? These lines by tight relationships, what I mean is that the scatter plot between the weight and the length are not randomly kind of scattered points. They seem to fall in a pattern, and that pattern is this kind of maybe a little bit curved, but generally positively trending line. But what you'll notice is that that line varies, the slope of that line varies by species, right? In some cases, like this striped bass, as you get longer, your weight increases pretty quickly. Whereas bluefish gets longer, but its weight increases relatively slowly comparatively with the striped bass. And so now we're able to very quickly, and I'm sorry that I probably, if I open this a little bit bigger, there's a lot we would need to cover about formatting these plots in a way that you could save them out better. And I think I should include some resources for that. But at least now, if I expand this, you can read all of the names of them. And if I saved it out like that, there we go. Make it a little bit smaller, then at least it's legible. And here I could save this image, say into, I'll just go through saving it, and this will be kind of the wrap up bit. I will save this into, I'll just save it into the data, and I will save it as fish weight length. I'm going to spell it right this time. And then I can go to data. So just to be clear again, you guys saw that I went export, save as image. I could also copy it if I just wanted to copy this plot and paste it into a Word document, for example. But I saved this, and now I'll go open it from here so you can see what I've done. We could write code to write out or save this plot directly from the console from R, but that would take a little bit more effort. And now I just want you to be comfortable generating the plots, and we can go through resources for you to save them out later. But there you go. Now you have the weight length relationship split out by species across the most commonly sampled species. So as is common with my self, when I generate this content, I get a little bit, maybe I'd say code happy. In here in the exercise, I should have just included this extra content as kind of the additional content for you to explore should you like, but I wrote this in as an exercise. So I stopped here at step 22. Step 23, I was shifting over to this fires data set and working through an example with plotting on the fires data set. It wasn't quite as long as what we just went through with the fish. But I would really encourage you to just use this existing exercise here in the next week to revisit this work and spend, probably take you 45 minutes, maybe, to come back and kick in a refresher and work through that on your own with a different data set and get some more practice with this plotting. Are there any questions here as we wrap up with the final 10 minutes? Where can we find the graphics again? I presume referring to the actual exercise piece. Let me come into here. So a little refresher of the Google Slides or Google Drive. Paste it again. I've just updated the Google Drive with this latest content. So you can come on to this link and go ahead and download this whole set of stuff. You might write over what you previously had. It will be very familiar. I mean, it will include all of the old stuff and some new stuff. So first of all, in the slides, you'll see, I don't know, this is just an extra file that doesn't mean anything. Each of the slides that we covered, so graphics goes over the slides. The exercises, you'll see the graphics exercise. That's word by word, like line, step by step, walking through all of this content. And then you'll see the data in this data folder, as well as the scripts if you want to. I can't look at it in Google Drive, but if you want to see the commented scripts, you have access to those as well. Great. So I guess I'll just wrap up by saying, I continue to feed the fed horse here, that this is, the tip of the iceberg with this material. I think that you'll get enough comfort and ability to get in to R and load your data and start exploring and start trying some of this work out, start developing some of those questions that you want to answer, and realizing maybe that you don't quite have enough, advanced knowledge to move forward. And that's when you'll have to respond to Sean or Laurel with ideas for future course offerings, or contact me and I can help you individually work through, what you want to do as well. But all of this, this is a huge world. This is kind of the peek into that with you guys, and hopefully some of you start to take this content and start to, explore and work further with it. I'd really like to, hear from some of you with your individual projects or problems and help work through what you're trying to explore and what you're trying to work on.